Hi, welcome to Dr. Medics. Kidneys, Part 2. Nephron. Nephrons are the structural and functional units of the kidney. Each kidney has 1 to 1.3 million of nephrons. Nephron starts decreasing after about 40 to 50 years of age at the rate of 0.8% to 1% every year. Each nephron is formed by two parts. One is the renal corpuscle or Malpighian corpuscle and the other one is the tubular portion, and it is also called as the renal tubule. Uriniferous tubule is divided into nephrons and collecting duct, and nephrons is divided into renal corpuscle and tubular portion. The renal corpuscle is divided into glomerulus and Bowman's capsule. Tubular portion is divided into proximal convoluted tubule, loop of Henle, and distal convoluted tubule. Loop of Henle is divided into three parts: descending limb, hairpin bend, ascending limb. Descending limb is divided into thick and thin, as well as the ascending limb is divided into thick and thin. Renal corpuscle or Malpighian corpuscle. It is a spherical and slightly flattened structure with a diameter of about 200 μ. Function of renal corpuscle is filtration of blood, which forms in the first phase of urine formation. Situation is renal corpuscle situated in the cortex of kidney, either near periphery or medulla. Classification of nephrons. Based on the situation of renal corpuscle, the nephrons are of two types: cortical nephrons or superficial nephrons and juxtamedullary nephrons. Classification of nephrons of the renal corpuscle. Based on the situation of renal corpuscle, the nephrons are of two types. One is cortical nephrons or superficial nephrons. Another one is the juxtamedullary nephrons. So the cortical nephrons present in the outer cortex near the periphery. This is the cortex. So the cortical nephrons present in the outer cortex near the periphery. In human kidneys, 85% of nephrons are of cortical nephrons. The function of the cortical nephron is the formation of urine. The second type of nephron is the juxtamedullary nephron. It is present in the inner cortex. Present in the inner cortex near the medullary or corticomedullary junction. Present in the inner cortex near the medullary or corticomedullary junction. So the function of juxtamedullary nephron is the concentration of urine and also formation of urine. The function of juxtamedullary is the concentration of urine and the formation of urine. Structure of renal corpuscle. The renal corpuscle has two portions, that is glomerulus and Bowman's capsule. So this is the glomerular and this blue one is the Bowman's capsule. The first portion is the glomerular. Glomerulus is the tuft of capillary enclosed in the Bowman's capsule. It consists of glomerulus capillary, which is interposed between the afferent arteriole, which is one side of the end, and the efferent arteriole, which is the other side of the end. This is afferent, efferent. Vascular system in glomerular is purely atrial system. Glomerular capillaries arise on the apparent atrial, divide into 4 to 5 large capillaries, each one subdivides into small mini capillaries arranged in irregular loops to form the anastomosis with efferent with efferent arterioles, which leaves the Bowman capsule. So the diameter of the efferent arteriole is actually less than the afferent arteriole. Glomerular capillaries are made up of single layer of endothelial cells. Single layer of endothelial cells which is attached to the basement membrane. These endothelial cells have many pores which are called as the fenestrae or filtration pores. On these diameter is 0.1 mu. The presence of fenestra is evidence of filtration function of the glomerulus. Second portion of the renal corpuscle is the Bowman's capsule. It is a capsular structure encloses the glomerulus, formed by the two layers, which is the inner is the visceral layer and the outer is the parietal layer. The inner layer covers the glomerular capillaries, continued with the parietal layer at the visceral pole. The parietal layer continued with the wall of tubular portion of nephron. Cleft-like space between the visceral and parietal layer continued as the lumen of tubular portion. Functional anatomy of Bowman's capsule resembles the funnel with filter paper. The diameter of Bowman's capsule is 200 mu. So now the functional histology is both layers of Bowman capsule are composed with single layer of epithelial cells on the basement membrane. The basement membrane of visceral layer fused with the basement membrane of glomerular capillary on which the capillary endothelial cells are arranged. And the basement membrane is fused together to form a separation between the glomerular capillary endothelium and epithelium visceral layer of Bowman's capsule. The basement membrane fused together to form separation between glomerular capillary endothelium and epithelium of visceral layer of Bowman's capsule. The endothelial cells of visceral layer fuse with basement membrane, but the fusion is incomplete. Each cell connected with the basement membrane by cytoplasmic extension of epithelial cells are called the pedicels. These pedicels are arranged in an interdigitating manner, leaving small cleft-like space in between. These called the slit pores. The epithelial cells with pedicel are called the podocytes. Tubular portion of nephron. The continuation of the Bowman capsule. It has three parts, which is the proximal convoluted tubule, loop of Henle, distal convoluted tubule. The first part is the proximal convoluted tubule. It is a coiled portion arising from the Bowman capsule, situated in the cortex. Continued as the descending limb of loop of Henle. The length is 55 mu. Proximal convoluted tube continued as the loop of Henle. Functional histology is proximal convoluted tube is formed by the single layer of cuboidal epithelial cells. The characteristic features of proximal convoluted tubule is the presence of hair-like projections directed towards the lumen of tubule. Because of the hair-like projection, the epithelial cells are called as the brush-bordered cells. Loop of Henle. It is divided into three parts: descending limb, hairpin bend, ascending limb. And now descending limb has two parts again. First one is the thick descending segment, and the second one is the thin descending segment. So the first is thick descending segment is direct continued of proximal convoluted tubule descends down into medulla in the length of 6 mm and diameter of 55 mu, formed by brush-bordered cuboidal epithelial cells. And now the thin descending segment continued as thin descending segment, formed by flattened epithelial cells without brush border, continued as hairpin bend of loop. Now the second part is hairpin bend, formed by flattened epithelial cells without brush border, continued as ascending limb of loop of Henle. Ascending limb or segment of Henle loop. It has two parts, one is the thin and thick. Now the thin ascending segment, continuation of hairpin bend lined by flattened epithelial cells without a brush border. The length of descending segment, hairpin bend and ascending segment is 10 mm to 15 mm. The diameter is 15 mu and continued at as thick ascending segment. Now the thick ascending segment. 9 mm length, diameter of 13 μ, lined by the cuboidal epithelial cells without brush border, terminal portion which runs between the afferent and efferent arterioles of the same nephron, which is called as the macula densa. And the macula densa part is of juxtaglomerular apparatus, ascends to the cortex and continues as the distal convoluted tubule. The last part of the tubular portion of nephron is the distal convoluted tubule. It is a continuation of the ascending segment and occupies the cortex of the kidney. So this is the distal convoluted tubule and this is a continuation of the ascending segment of and occupies the cortex of the kidney. Continued at collecting duct. Continued at collecting duct. The length of the distal convoluted tubule is 14.5 to 15 millimeter. The diameter is 22 to 50 mu. The functional histology of distal convoluted tubule is lined by the single layer of cuboidal epithelial cells. Cuboidal epithelial cells with brush border, and the epithelial cells are called the intercalated cells. This is called the eye cells. Collecting duct is the apart from the uriniferous tubules. The uriniferous tubules have nephron and the collecting duct. Before all the things what we have learned is a part of nephron. The collecting duct is a part of uriniferous tubule. Distal convoluted tubule continued as a primary or arch collecting duct. Arch collecting duct, which is in the cortex of the kidney. Lower part lies in the medulla. 7 to 10 primary collecting ducts unite to form straight collecting duct which passes through the medulla. The diameter is 400 to 200 μm. The length of collecting duct is 20 to 22 millimeters. The collecting duct is formed by the cuboidal or columnar epithelial cells. The functional histology. The collecting ducts are two types of epithelial cells. One is the T cells, which is the principal cells, and I cells, which is the intercalated cells. The passage of urine. At the inner zone of medulla, it has a straight collecting duct from each medullary pyramid unite to form papillary duct or duct of Bellini, open into a V-shaped area called the papilla. Urine from the medullary pyramid are collected in the papilla, which is drained into the minor calyces, and 3/4 of minor calyces unite to form major calyces. One major calyces is here. Major calyces. Each kidney has 8 minor calyces and 2 to 3 major calyces. So urine in the minor calyces passes through the major calyces and opens into the pelvis of ureter. This is the pelvis of ureter and passes through the ureter and reaches the urinary bladder. Urinary bladder is emptied through urethra.